Hello everyone, and welcome to our lecture for Chapter 9, covering microbial metabolism. As always, you have your lesson objectives. If you have any questions about how to use those best, let me know, but I figure by this point in the semester, you probably have figured out how best to use those for you. So to start things off, we first want to talk about what metabolism is. A lot of times we think we understand this term because we hear it so often in the media, online somewhere, through just general conversations. But when we get down to it, a lot of us really don't know what metabolism actually is. So metabolism, this term, is derived from a Greek word that means change. And when we use this term in a cellular context, what we are talking about are all of the chemical and physical changes that occur inside a cell to allow it to survive. Maybe those physical and chemical changes occur so that it can process nutrients and use those to make cellular structures, or maybe it has something to do with using energy, or any given activity that a cell undergoes, if it's some kind of physical or chemical change, is part of metabolism. For metabolism to occur, there are certain materials that are required and certain conditions that need to be met. We are going to focus on the first two materials and conditions in this lecture, and then focus on the third one in the next lecture. So for metabolism to occur, there are three main things that have to be present. There have to be enzymes present, there has to be energy present, and there have to be raw materials and nutrients present. In this lecture, we'll talk primarily about enzymes and energy, and then in the Chapter 10 lecture, we will talk about nutrients and how those are used to help keep cells alive. So before we launch into what enzymes and energy do for cells or what they do for metabolism, we first want to talk about a couple of different types of metabolism. There are two very broad divisions of metabolic reactions, and those are catabolic reactions and anabolic reactions. Catabolic reactions are used to break things down. So catabolic reactions take large substances and break them into smaller ones. Basically, a bunch of decomposition reactions occur that take something very large and break it into smaller pieces. Typically, in the process of breaking something large into small pieces, energy is released. Anabolic reactions are the opposite of these. These are reactions that build things up. So anabolic reactions take small substances and join them together through synthesis reactions to create some kind of larger structure. Most of the time, these reactions consume energy, so they require energy to happen. One thing I do want to point out about metabolism in general is that a lot of times people can find this to be a very dry subject. It can be kind of confusing in certain situations, but please keep in mind the big picture of this. And the big picture of this is that microbial metabolism is really one of the main forces that controls everything in the world. Think back to Chapter 1 when we were talking about things like photosynthesis, and remember that microbial photosynthesis is really the thing that allowed the atmosphere of the Earth as we know it to develop. And had those microbes not developed this metabolic process, we would not be able to exist, at least in this current form. So microbial metabolism has the power to change the world. It has the power to change someone's body. It has the power to make plants grow or make plants die. It really is kind of the basis of making sure the rest of life functions. And so keeping that in mind, I hope that you will find this to be a little bit more interesting than you may have found in previous exposures to it, and that you can at least appreciate why this is such an important thing to talk about both for just your own personal well-being and understanding, and also in the healthcare professions. Because when someone becomes infected by a bacterium or some other type of microbe, that organism goes through metabolism, and those processes can damage the human body quite a bit. So understanding how microbial metabolism affects all of us as a whole as a species, as well as each of us individually, is really important in making sure we stay alive and stay healthy. One of the main things that is required for metabolic reactions to occur is the presence of enzymes. Enzymes are protein molecules that assist with metabolic reactions. They're oftentimes referred to as catalysts because they help chemical reactions occur, but they themselves are not changed in the process. They're not used up or altered or anything like that. In the metabolic world, enzymes are kind of like a matchmaking service or some kind of dating app. They basically can help bring things together, they can help certain reactions occur, but they themselves are not really involved in the process or used up in that regard. Mostly what enzymes do is help lower the amount of energy that is required for a chemical reaction to take place. And they can do this through a few mechanisms. First, they might just bring substances closer together. So they might bring what we call the reactants closer together to allow them to combine. And by bringing them closer together, they may help create this physical space for the chemical reaction to occur. So it may be that two chemicals would be able to combine with each other, but they need a space to be able to do that, and the enzyme might help provide that space. Without that space, these two chemicals can't combine or can't split up, but with that space that the enzyme provides, they're able to go through their reaction. Sometimes they can help change the shape of some type of reactant or molecule. Basically, what the enzyme can do is kind of attach to this reactant or these reactants and alter their shape a little bit so a chemical reaction can take place. They also may help speed up reactions. Sometimes there's a situation where a chemical reaction could occur, but it would happen so very slowly, it would take maybe months or years or decades to occur, and a cell doesn't have that long, it can't wait that long. So the enzyme actually allows this process to take place at a much faster rate. So those are kind of all of the things that enzymes do to allow metabolic reactions to happen. They bring reactants closer together and help create a physical location or physical space for this to take place. They may change the shape of the reactions, or they speed things up so this reaction can actually occur and the cell can stay alive. Enzymes are used in both catabolic and anabolic reactions. Typically, we start with what is called a substrate and end with what is called products. So we start with a substrate, or they're also called reactants, whether there's one or two or three or four. It's just called the substrate or the reactants. And then at the end of the enzymatic reaction, we have products or a product. And another thing about enzymes is that, especially with bacteria, they are often used either inside the cell or outside the cell. So sometimes bacteria will produce an enzyme and it uses it just in its cytoplasm, but other times a bacterium will produce an enzyme and it will excrete that enzyme into the external environment so it can break down something large and bring it into the cell so that it can use those parts. Some enzymes work perfectly well on their own and they're able to catalyze the reactions that they are designed to without any other help or assistance, but other enzymes may require some other types of molecules to be able to function the way they need to. Cofactors and coenzymes oftentimes provide some assistance to enzymes to ensure that the metabolic reaction that they need to catalyze will actually be able to take place. Cofactors are typically inorganic molecules. They're not always inorganic, but oftentimes they are, and oftentimes they are metals. Metals are usually very good at grabbing onto electrons and passing them on to other substances. And so oftentimes cofactors are joined to an enzyme to help increase the rate of the chemical reaction that it's supposed to catalyze. So these metals that oftentimes will be added to an enzyme will grab onto electrons from one molecule and pass them off to another, and this will kind of speed up the rate at which the reaction occurs. Coenzymes are typically organic molecules, and oftentimes they will kind of alter the shape of the enzyme to allow a substrate to fit better. So most of the time a coenzyme will attach itself to the active site of an enzyme. So for example, maybe this coenzyme attaches itself right here and just allows this substrate, this molecule B, to fit a little bit better in the active site so that that chemical reaction can occur. The second main thing that needs to be present for metabolic reactions to proceed is energy. And energy is another concept that most of us think we understand fairly well, but just to make sure we're on the same page, we're going to go ahead and define it right here before we get started talking about how it works and how cells go about transforming it into a form that works for them. So energy is the ability to do work or cause change. Basically, this is the power that cells use for metabolism. And all organisms need a constant input of usable energy from their environment. So one of the things to remember is that there is energy all around us, all around microbes, all around everything in the world and in the universe really, but not all of it is usable by every type of organism. So one example that is really great to illustrate this is that when you go outside and the sun is shining, there is a lot of energy coming from the sun. There's a lot of light energy that's present in the environment. However, to you as an organism, that is not usable energy, at least not directly, because you do not have the ability to take that energy that's coming from the sun and directly transform it into ATP. So you can't do that. You have to rely on other organisms like microbes and plants to harvest that energy from the sun, take that energy and turn it into chemical energy, and then you could eat those plants or potentially those microbes and gather the energy in that form. So all organisms have to have energy input from their environment, and that energy has to be in a way that they can use it. So when we are talking about energy or considering the types of energy that a certain type of microbe needs to have available to it, we look at it in kind of three main ways. We look at the environmental source of energy, whether that microbe needs chemicals as energy or can use light as an energy source, how that microbe is going to convert that energy in the cell. Because all organisms, whether they are microbes, humans, plants, animals, fungi, etc., all of them are going to take the energy from their environment and convert it into a different form. No organisms just use basically raw energy from the environment. All of them convert it in some way or another. And the end product of converting that energy is typically ATP. There are some other options that are available to organisms, but the most common thing to do with energy from the environment is to create ATP. So when we think about energy in terms of metabolism, there are these three main factors to consider. One, what is the environmental source? Is that source chemicals or is it light? Two, how is that microbe or that cell going to convert the energy that it got from the environment? And in that process, how is it going to move electrons through its body? And three, what is the end product going to be? And typically, like I said, that's going to be ATP, although there are some other options. Now, our next job is talking specifically about ATP production, how exactly that is done. Because one of the main purposes of metabolism is to take energy from the environment and use enzymes to convert that energy and turn it into ATP. So those are the reasons that I've been talking about enzymes and talking about energy and what it is, and trying to relate it back to microbial metabolism as a whole. So ATP production is a big deal for organisms because ATP is actually the chemical source of energy that is used to power metabolism. So all organisms need energy that they get from their environment. But like I said, organisms don't just take that raw energy they get from their environment and use it inside their cells to power metabolism. First, they convert that energy into some kind of chemical, which is typically ATP. And then they use the energy that's stored in the ATP to power the rest of their metabolic reactions. So all organisms, no matter what type they are, need a few basic things to go through ATP production. First, they need an electron donor. Electron donors are inorganic or organic chemicals that will release electrons. So here over on my pencil isn't working right now, but over on our diagram here on kind of the upper left-hand side, we have a donor. That's our electron donor. They also need an electron acceptor. They need something to grab onto those electrons that the donor is going to give away. Electron acceptors are also inorganic or organic molecules, and they will receive the electrons. They also require raw materials. A lot of times these raw materials are things like phosphate groups, hydrogens, water, something we call ADP, which is adenosine diphosphate, because that is going to actually get turned into adenosine triphosphate, the ATP. And the basic kind of big picture mechanism of how this all works is that the electron donor comes from somewhere in the environment and it donates electrons to various electron acceptors throughout the cell. As those electrons are transferred from the initial donor to the acceptors, energy moves throughout the cell. Another way to think about energy besides the power to do work, at least in this context, is the flow of electrons. So our electron donor starts the flow of electrons by giving electrons to the first electron acceptor. And then depending on what type of organism we're talking about, depending on what type of process we're talking about, that electron acceptor might turn around and become an electron donor and give electrons to another molecule, which will then give electrons to another molecule, etc. So this initial electron donor starts this flow of electrons through the cell, which is essentially a flow of energy through the cell. That energy typically then ends up getting used to power the process of sticking a phosphate group onto what we call ADP, adenosine diphosphate, and creating ATP. At the end, this whole process, there's ATP and then waste products. So that is the big picture process of ATP production. It starts with some kind of electron donor, typically that's obtained from the environment, and that electron donor starts creating this flow of electrons or this flow of energy through the cell by donating the electrons. Other molecules accept the electrons and pass them off, and this transfers energy throughout the cell until that energy can be used to take a phosphate group and stick it on to adenosine diphosphate to create ATP and waste products. Then the energy that's contained in ATP can be used to power the rest of metabolism, whether that is some type of synthesis reaction to build up a cell wall, or go through DNA replication, or it's a decomposition reaction to break down some type of waste product and get rid of it. There's lots of different things that a cell could be doing with this energy that is now stored in ATP. Microbes have many different pathways that can be used to produce ATP. So there are lots of different ATP production pathways that exist in the world. However, we are going to focus on just 4 of the most important or 4 of the most common pathways out there. We are going to look at photosynthesis, aerobic respiration, anaerobic respiration, and fermentation. So even though there are lots of different ways aside from these 4 ones to make ATP, we don't necessarily have time to go into each of those, and we're just going to focus on these 4 very common pathways. For each of these, I have the electron donor, the final electron acceptor, and the waste product written out. We are going to go into each of these in detail, but you may want to keep this slide handy so that you can refer to it while we're looking at the details of each of these. So basically with photosynthesis, the initial electron donor is water, and the final electron acceptor is what we call NADP+. The waste product, at least in the type of photosynthesis we're talking about, is going to be oxygen. For aerobic respiration, anaerobic respiration, and fermentation, we are going to use glucose as an electron donor and look at what happens when glucose is used for that purpose or in that capacity, even though there are other electron donors that could start this process out. We'll also look at the final electron acceptors for each of these and the waste products that are produced. Our first ATP production pathway that we're going to look at is photosynthesis. Now, one thing that's important to understand is that there are different types of photosynthesis. We are going to focus on what's called oxygenic photosynthesis, which is the type of photosynthesis that produces oxygen as a waste product. But there are different types. What all of them have in common is that each of them uses the energy from light to start that flow of electrons I was referring to earlier. So step 1 of this process is that light energy comes in from the environment and hits a pigment that is contained in the microbe. The transfer of this energy from the light to the pigment, which is a chemical in the microbe, causes a molecule of water to be split in half. So water, that chemical formula is 2 hydrogens and 1 oxygen. And this energy that came in from the light is high enough that or is in great enough amount that it allows that molecule of water to be split in half, and electrons are released as a result. So those electrons are going to get passed through a series of molecules called electron carriers. You do not need to memorize the names of those electron carriers. That is not relevant to this course at this point in time. What you want to be focusing on is the fact that the energy from the sun or some other light source has been transferred from the environment to the microbe. That energy has been used to split water, free electrons, and those electrons have started to flow through the microbe. At the same time that flow of electrons starts and those electrons are getting passed through the carriers, the oxygen as a waste product is actually produced and some free hydrogen is produced. So those are kind of hanging out in the cell now. The electrons continue to flow through the carriers, and as this process continues to happen, more and more molecules of water are split, and so more and more hydrogen accumulates in the cell. The oxygen that is created ends up just being released as a gas, but the hydrogen ions end up staying here in the cell. And those hydrogen ions end up taking place in 2 different reactions. Some of them combine with a molecule called NADP+. NADP+ is an organic molecule that is common to many types of organisms, and when it combines with a hydrogen ion, it forms what's called NADPH. So NADP+ basically accepts some of the electrons that were generated when water was split. So it grabs onto those electrons and essentially ends the flow of electrons through the cell. Other hydrogen ions are going to interact with an enzyme called ATP synthase. ATP synthase is a protein that's kind of tube-like or kind of like a straw. It has a hollowed out center and it sits in the cell membrane of certain types of microbes. And its job is to help turn ADP into ATP. So to do this, hydrogen ions come through the center of this tube-like protein, and they provide some energy that enables this enzyme to take a free phosphate group and stick it on to ADP to create ATP. So basically what the cell has done here is taken the energy from light and captured it in a pigment. That pigment has then used that energy to split apart water and generate some oxygen, some hydrogen, and a flow of electrons. Those electrons flow through the cell and transfer energy as a result through these carriers, and they end up at the end attaching to NADP+. That creates a molecule of what we call NADPH. Other hydrogens that are hanging around, some of which were generated during the splitting of water, will come over to ATP synthase and will allow this enzyme to turn ADP into ATP. So now that energy that ultimately came from light has been transformed. It's been moved around the cell and turned into a chemical energy that is being held in this molecule of ATP. So that energy from light ultimately got turned into chemical energy and is now hanging out here in ATP. The cell can then use this energy that's hanging out in ATP to do all kinds of other metabolic reactions. Now, the good news about this slide is I'm actually not going to test you very much on photosynthesis. The only types of questions you might run across on an exam or quiz about photosynthesis are what the initial light source is, what the waste products are, whether ATP is generated, etc. I'm not going to test you on the names of any of these characters, and I'm not even really going to ask you to explain how light energy gets turned into chemical energy. I use this to help you get used to this idea of the flow of electrons, introduce some vocabulary terms and some concepts so that it eases you in to some of the other topics that you are going to be tested on. So now we get into respiration and fermentation, and these are processes you will be tested on. The reason for that is most of the organisms that infect people and infect animals go through these processes. There are very few organisms on the planet that use photosynthesis that could actually infect a human or an animal. The reason for that being they need light as a source of energy, and humans and animals are not good sources of light at all. So basically, our bodies do not provide the right types of environments for organisms that use photosynthesis to survive. However, respiration and fermentation use chemicals to get their metabolism started, and humans and animals happen to be excellent sources of chemicals. So these are very important metabolic processes for future healthcare providers to understand, because the organisms that infect humans and the organisms that infect other animals typically use respiration or fermentation to go through metabolism to generate their ATP. And as a result, these processes are some of the things we target when we're trying to control infections. So, all that being said, it is time to go ahead and launch into the details of these processes, starting with respiration first. There are two types of respiration. There's aerobic respiration, which uses O2 or oxygen as a final electron acceptor. And there's anaerobic respiration, which uses an inorganic molecule that is not oxygen as an electron acceptor. In some classes, you may have been taught that the process that's actually fermentation is anaerobic respiration. And I am sorry for that. That's very common in human-based biology classes, things like human biology, anatomy and physiology sometimes. They will often call the process that is actually fermentation, anaerobic respiration. So if you have learned that in another class, you're going to have to unlearn that terminology, because in microbiology, there is a difference between anaerobic respiration and fermentation. So, aerobic respiration and anaerobic respiration both use 3 stages to create ATP. The first stage is called glycolysis. During glycolysis, a molecule of glucose, which is an organic chemical, is split up into multiple parts. This process of splitting glucose essentially is used to free up electrons and reposition electrons that are going to later be used to create ATP. So the big picture purpose of glycolysis, of splitting up glucose into smaller pieces and creating what we call pyruvate, is to free up some electrons, put them in a different position, and get them ready to be used to create ATP. Basically, it's taking the energy in glucose and rearranging it so it's starting to get ready to have that energy that's contained in the glucose molecule and get it put into ATP. At the end of glycolysis, there are 2 molecules of something called pyruvate. It's also sometimes called pyruvic acid. It's the same basic thing. Don't worry about the difference. You can use those terms interchangeably. Pyruvate is used to start the next step, which is the Krebs cycle, also called the citric acid cycle, or sometimes the tricarboxylic acid cycle. All 3 of those things mean the same thing. So I tend to say Krebs cycle just because that was very common when I was being taught about this process, but I think a lot of books are switching to citric acid cycle more commonly. It's the same thing. So the pyruvate that was created when glucose was split into pieces goes into the citric acid cycle. The purpose of this process is to rearrange that energy that was contained in glucose and that got put into pyruvate a little bit further and create what we call NADH and FADH2. NADH and FADH2 are complex organic molecules that are rich in electrons. They have a lot of electrons. So these molecules take the electrons that were in glucose and they're going to get them ready to go to the final stage of respiration, which is the electron transport chain. So the purpose of glycolysis is to split up glucose and create pyruvate and free up some of that energy. The purpose of the pyruvate is to go into the Krebs cycle, rearrange that energy a little bit further, and put that energy into what we call NADH and FADH2. NADH and FADH2 are then going to go to the electron transport chain, which is the final stage of both aerobic and anaerobic respiration. What happens here is that the NADH and FADH2 donate electrons and lose some hydrogens in the process. So the electrons from NADH and FADH2 go to electron carriers. So the electron carriers are going to pass those electrons on to each other, just kind of in a sequence. You don't need to worry about the names or the numbers or anything like that. You're just going to call them electron carriers. And at the same time that those electrons are flowing through these carriers, hydrogens are accumulating. Once the electrons reach the last electron carrier in the cell membrane here, they're going to combine with oxygen. This is going to create water. So the purpose of oxygen is to be the final electron acceptor in this process, grab onto those electrons, and as oxygen grabs onto those electrons, it's also going to grab some hydrogens and create water. Any hydrogens that are not grabbed onto by oxygen are going to be used to help ATP synthase create ATP. So very similar to what we were talking about in photosynthesis, ATP synthase is, again, a tubular protein. It's kind of shaped like a straw. It has a hollow center that hydrogens can pass through. As these hydrogens pass through this ATP synthase, it causes the phosphorylation of ATP. And what that means is that a phosphate group is stuck on to ADP, and that creates ATP. To phosphorylate something means to stick a phosphate group onto something. And in this case, the something that we're sticking a phosphate group onto is an ADP, an adenosine diphosphate. So if we take an adenosine diphosphate and stick a phosphate group onto that, that makes adenosine triphosphate. And now the energy that was once contained in a glucose molecule has been moved to ATP, and that ATP can be used to power the rest of the metabolic reactions for the cell. So I've explained basically the process of aerobic respiration and talked about how there's an oxygen waiting at the end here. And the process of anaerobic respiration is exactly the same, except instead of oxygen being waiting there at the end, there's some other kind of inorganic molecule. There are lots of different options. It could be something like a sulfate, it could be a nitrate. You don't need to worry too much about exactly what could be there. What you need to focus on is that it is an inorganic molecule that is not oxygen. So some organisms will use oxygen as the final electron acceptor. Others may use another type of inorganic molecule that is not oxygen. But either way, whether it is oxygen down here or a different type of inorganic molecule, all three of these processes are the same. Glycolysis occurs in the cytoplasm of the cell. The Krebs cycle also occurs in the cytoplasm of the cell. The electron transport chain is found in the cell membrane. And whether it is aerobic or anaerobic respiration really is just determined by what the final electron acceptor is here. So all these electron carriers are pretty much the same. It's just if there's oxygen waiting here, it's aerobic respiration. If it's some other type of inorganic molecule, it's anaerobic respiration. Now, I know this was a lot of information to go over, and this diagram's a little bit small. So on the next slide, I have this diagram blown up a little bit, and I'm going to go over it again just one more time to help it solidify in your brain. Aerobic and anaerobic respiration have 3 stages. The first stage is called glycolysis. This happens in the cytoplasm of the cell. And the purpose of glycolysis is to take glucose, an organic molecule with a lot of energy stored in it, and split it up into 2 main pieces called pyruvate. Splitting up this glucose allows the energy, or the electrons, that are contained within this molecule to start to be freed up. They're not quite ready to be used to make ATP just yet, but they're getting in position. They're getting ready to make ATP. Pyruvate then is used to power the Krebs cycle. So pyruvate goes through this process of a series of chemical reactions, and as a result of these various chemical reactions, NADH and FADH2 are produced. These molecules are very rich in electrons. They have a lot of electrons. Some of those electrons came from glucose. So we're starting to see how the energy from glucose is going to directly get used to make ATP. So NADH and FADH2 that are rich in electrons are going to migrate to the cell membrane. In the cell membrane, there are various molecules. They are metals, they are proteins, sometimes a combination of both, that are called electron carriers. NADH and FADH2 are going to donate some electrons and give them to these electron carriers. When they give those electrons to these electron carriers, they also free up some hydrogens. The electrons migrate through these electron carriers, and once they reach the final one that's sitting in the cell membrane, they have to go somewhere. So they are either going to attach to oxygen, if it happens to be aerobic respiration, or some other kind of inorganic molecule, if it's anaerobic respiration. If we're talking about oxygen being here and this process being aerobic respiration, then this is the place where water is created as a waste product. If it's anaerobic respiration and there's something other than oxygen here, typically what's created is some kind of acid as a waste product. Now, while that is going on, while these electrons are migrating through carriers and they're reacting with their final electron acceptor to either create water in the case of aerobic respiration or some kind of acid in the case of anaerobic respiration, there are hydrogens that have been accumulating near this ATP synthase. ATP synthase is an enzyme that allows for the phosphorylation of ADP to create ATP. Hydrogens pass through this enzyme. Remember, this enzyme is kind of tube-like in nature. It's kind of like a straw. It has a hollowed out center. And as those hydrogens pass through this ATP synthase, they provide some energy for the ATP synthase to take a phosphate group and stick it onto ADP. Basically, they allow for the phosphorylation of ADP, which creates ATP. So now we have energy that was once contained in glucose and has traveled through glycolysis, the Krebs cycle, and the electron transport chain, and finally made it all the way to ATP. The energy that's stored within ATP can now be used to power the rest of the cell's metabolism. It can go ahead and use it for synthesis reactions, it can use it for decomposition reactions if it needs it. Either way, whatever it needs to do, now the cell has energy in a usable form to carry out the metabolic reactions it needs to survive. So far, we've talked about 3 out of the 4 most common ways for microbes to make ATP. Remember, there are more than just this. However, we are focusing on the 4 most common ones, and out of those 4, 3 of the most common ones used by microbes that infect people and animals. So far we've talked about photosynthesis, which is obviously important for creating oxygen, but it's not really used by organisms that infect animals. We've also talked about aerobic and anaerobic respiration, which are almost exactly the same process, the only difference being that final electron acceptor that's found in the electron transport chain. Now we're going to talk about fermentation. This pathway is sometimes called anaerobic respiration, but it is not. So it is very common for human biology-based classes, whether it's human biology or anatomy and physiology, or some other kind of human-based course, to call this process anaerobic respiration. It's really important that if you learned that terminology, that you undo that now, because there are some major differences between what is true anaerobic respiration and what is actually fermentation. So respiration, whether it is aerobic or anaerobic, uses 3 stages to create ATP: glycolysis, the Krebs cycle, and the electron transport chain. Fermentation, though, is basically just using glycolysis to create ATP. It is an anaerobic process, meaning that it happens in the absence of oxygen, but it is not respiration because it doesn't use the Krebs cycle, it doesn't use an electron transport chain, and it doesn't use this flow of electrons that's found through the carriers to create ATP. Another big difference is that in respiration, inorganic electron acceptors are used. But in fermentation, organic electron acceptors are used. So that's another key difference between respiration and fermentation. And again, like I've mentioned previously, respiration uses the Krebs cycle and the electron transport chain in conjunction with glycolysis, but fermentation just uses glycolysis. There are multiple types of fermentation out there. We are just going to go into a very broad, kind of big picture overview of fermentation as a whole. And when we look at fermentation from a big picture perspective, there are two main types. There's a type that produces alcohol as a waste product, and a type that produces acid as a waste product. The type of fermentation that produces acids as a waste product typically produces something called lactic acid. This type of fermentation can happen in a lot of different organisms, including humans. And in this process, what happens is that glycolysis will be used to split up glucose and create pyruvic acid, or pyruvate. In the process, some ATP is created and some NADH is created as well. From there, NADH reduces pyruvate or pyruvic acid to create lactic acid. So basically, in this process, glucose has served as the initial electron donor. Pyruvate is the final electron acceptor, and that's an organic molecule. And then pyruvate has to get reduced into some kind of waste product, so lactic acid or lactate is created. Many types of bacteria use this method of ATP production either preferentially, meaning they prefer to do that over something like aerobic or anaerobic respiration, or some of them will use it if they don't have the means to carry out aerobic or anaerobic respiration. One example of where this can happen is in the human mouth. We've talked about the biofilm that forms in the human mouth before. And if we don't take care of that biofilm, it tends to build up. And the bacteria at the base of that biofilm will go through this process of fermentation. So when we put food into our mouths, especially sugars, they are able to use those sugars to go through this process of fermentation and create lactic acid that eventually can start to wear away our teeth and create holes in them. This is not the only place that this type of fermentation can happen. It's just an example that a lot of people will be able to understand and kind of conceptualize. The other type of fermentation is a type that produces alcohol, and a type of alcohol that is frequently produced is called ethanol. So our second type of fermentation that we'll talk about is ethanolic fermentation. And this process starts out in a very similar manner to lactic acid fermentation. Glucose is split apart during the process of glycolysis, and pyruvate is produced as a result. NADH is also produced, as well as some ATP. In this particular situation, though, instead of pyruvate being used to create lactate, CO2 gets released from the pyruvic acid, and the NADH is then used to combine with that pyruvic acid to create ethanol instead of lactic acid. So it's a very similar process. The final waste product, which is ethanol, is different, but overall they're very similar in that they both use glucose as the electron donor. Glycolysis is the only metabolic pathway that's used to create the ATP. And then pyruvate, that organic molecule, serves as a final electron acceptor, and it is used or broken down in some way or another to create a waste product. This type of fermentation is very commonly carried out by certain types of bacteria as well as yeast, which are single-celled fungi. And an example of bread rising is an example of ethanolic fermentation. So we don't taste the alcohol in the bread because it's, one, a very small amount that's produced, and two, it basically evaporates during the cooking process. But for yeast to be able to create the rising effect in bread, they are going through this fermentation process. They're releasing CO2, which is a gas which causes the rising, and producing some alcohol as a waste product as well. Our last slide in this lecture is just a little bit about electron donors themselves. For the most part, we have focused on glucose as an electron donor. So with aerobic respiration, anaerobic respiration, and fermentation, we've looked at what happens when glucose is used as the electron donor. But one thing I do want you to understand is that there are lots of different types of electron donors that microbes are very good at using. And sometimes they will use something other than glucose or something other than other types of sugars to create ATP as well. Many microbes are very good at using something like a protein or a lipid as an electron donor as well. Carbohydrates are typically preferred by most microbes to create ATP because they provide the most ATP when going through these metabolic processes. But in their absence, other molecules can be used. And two of the most common molecules to be used, especially by microbes that infect people and animals, are lipids and proteins. So for an organism to be able to use a lipid as an electron donor, it has to have an enzyme called a lipase. A lipase is an enzyme that will break lipids into fatty acids and glycerol. And from there, the organism is able to use those molecules as electron donors to start this ATP production pathway. This is just to give you an example of when this can happen. There is a bacterium called Staphylococcus aureus. If you haven't heard of it yet, don't worry, we're going to talk about it quite a bit in the disease lecture. But many people have already heard about this type of microbe. A lot of times people just call it staph. And this microbe is very, very diverse in terms of what it can use for its electron donors and where it can live in the human body. And if it gets into our eyes, which oftentimes have some associated oils to help keep the skin there lubricated and everything functioning the way it's supposed to be, it will often, instead of using carbohydrates as an electron donor, start to use the lipids. In this situation, this condition is called blepharitis, which is also just kind of a fun word to say, which is part of why I use this as an example. But basically what happens here is the bacteria start using the lipids on and around our eye tissue to create their ATP and start infecting the eyelid and nearby areas. So that's just an example of what can happen in the human body if there aren't sugars around for microbes to use. Some of them can just switch. Another common type of electron donor for microbes to use if there are not carbohydrates or sugars around are proteins. The human body and animal bodies are very rich in proteins, so if a microbe can't access a sugar, then it could switch to a protein, and that's not such a big deal for it. To use proteins as electron donors, microbes have to use an enzyme called proteases. The proteases break these proteins down into individual amino acids, and then those individual amino acids can be kind of modified to be able to be used as electron donors. There isn't a specific example I have of this in the human body or in an animal body because it just can happen all the time. But basically, if a microbe infects a human or animal and there's not a sugar around nearby and maybe not a lipid, they can very easily just use proteins that are in our tissues or proteins that are being used to hold our cells together, like connective tissue, and just start breaking those down and using those as a source of materials to create ATP.